Hi everyone, this is Lilith, and I'm one of your IAs for BIBC 102. And in this video, I will quickly go over Lecture 6 and Lecture 7. To begin, a small definition for Warburg effect. The Warburg effect basically explains the ability of tumor cells to take up large amounts of glucose and turn it into large amounts of lactic acid by performing glycolysis, which is a type of anaerobic respiration, meaning that it does not require oxygen to take place. The next concept we discussed in lecture was what happens to pyruvate after it's been made, and this is the structure of pyruvate for your viewing. And some things that Dr. Hampton explained in class were that pyruvate, after it is made through glycolysis, can go either into ethanol production or into lactate production, or it can be shuffled into the mitochondrion to enter the citric acid cycle or the Krebs cycle, which we will learn later in this video, and to be further broken down into carbon dioxide and water. So I'll quickly go over each ethanol production and lactate production on this side over here. So in both reactions, or I guess in both pathways, making lactate and making ethanol, we will be regenerating the molecule of NAD over here and over here. And why is this good? If you remember back to glycolysis, we used NAD plus molecule, right, to make NADH. Now, if you think about it, if NAD plus is never regenerated, then we will get stuck with a lot of NADHs, correct? And if there's no more NAD, then glycolysis would not be able to take place anymore. So glycolysis will be halted if there's no more NAD+. That's why cells have, that's one of the reasons to why we have these pathways, because they actually use up NADH plus H plus to make NAD, so that NAD can go back and be used in glycolysis, so that glycolysis pathway does not stop. So now that that is out of the way, let's look at how pyruvate turns into lactate. Our pyruvate that has freshly come out of glycolysis will be taken up by lactate dehydrogenase, which is the enzyme that uses NADH plus H plus to reduce this carbonyl over here into an alcohol. And this structure is L-lactate. Very simple reaction. Lactate or lactic acid is colloquially known as the thing that's produced in your muscles after you do a lot of anaerobic exercise, and it causes muscle ache. But I believe that has been debunked. It actually isn't the levels of lactate that cause that pain, but it is true that a lot of lactate is produced when we do a lot of anaerobic exercise, for example, weightlifting or doing short sprints. The next pathway, pyruvate turning into alcohol. This is a little bit of a longer pathway, but still not too complicated. We have our pyruvate, and pyruvate is worked up by this enzyme called pyruvate decarboxylase. And the name itself kind of tells you about what this enzyme does. Decarboxylase, meaning that this enzyme will take off a carboxyl group from pyruvate, and that's exactly what it does. We should probably talk more deeply about this enzyme because it uses the cofactor TPP, thiamine pyrophosphate, and let's look at that over here. Basically, what TPP does is it attacks carbonyls, right? And I've made this small thing. Hopefully it helps you remember that TPP attacks carbonyls. And how does it do that? It has this business end that has a carbanion ion, and a carbanion ion is a carbon molecule over here that has a negative charge. And as you know, negatively charged carbon atom, not molecule. Negatively charged atoms are very good nucleophiles, and what they do is they attack this carbonyl. This carbonyl bond opens up, And as you can imagine, a little bit of OChem, many reactions take place after this opened up carbonyl bond closes, and then this is kicked off. And as you can see, this is carbon, oxygen, oxygen. That's exactly CO2. So what happens is CO2 is kicked off, hence the name decarboxylase. And what we are left with is this portion of pyruvate, which is called acetaldehyde, right here. So with this step, we form acetaldehyde, and the next enzyme in the next step is alcohol dehydrogenase. And as I've said before in my previous videos, every time you see a dehydrogenase, think that a reduction or an oxidation is taking place. And staying true to that, we indeed do have another NAD being produced from NADH usage. So what happens here specifically, NADH is used to reduce this carbonyl over here into an alcohol. And as you may know, an OH group on 2 carbons is called ethanol. And that's exactly how ethanol is made from pyruvate. So now that we have gone over the 2 anaerobic pathways of where pyruvate could go, right? Lactate production and ethanol production. In the next coming slides, we will talk about the Krebs cycle and which is the aerobic pathway that pyruvate takes. But before we get there, we need to talk a little bit about regulation in glycolysis. Let me see what the next page is. Then we'll talk about the pentose phosphate pathway, and then we'll talk about the Krebs cycle. Okay, a little bit of preview for you there. Okay, so we talked about regulation in glycolysis, and this is very important because glycolysis is a pathway that has many intermediates, right? It produces the molecule pyruvate that can go into other cellular pathways, and the cell needs a way to regulate the important steps of glycolysis, or else the cell will have absolutely no control of this pathway and won't be able to use it to its advantage. So the 3 enzymes that are very important and release a lot of energy during glycolysis are hexokinase, phosphofructokinase 1, and pyruvate kinase. And these are the sites of allosteric regulation, and they are important enough for us to take a closer look. So hexokinase, also known as glucokinase if this enzyme is in the liver. Glucokinase is simply an isozyme of hexokinase, and it only exists in the liver. So hexokinase and glucokinase are regulated allosterically by glucose 6-phosphate. And if you think about it, this makes sense because glucose 6-phosphate is the product of the hexokinase enzyme. So if a lot of glucose 6-phosphate is produced, then glucose 6-phosphate will negatively feed back onto hexokinase, telling it to stop being very active because there's a lot of this, right? And if there's not enough of glucose 6-phosphate, then it will activate allosterically and tell hexokinase to produce more of this. So by using allostery on hexokinase, glucose 6-phosphate is able to regulate how active hexokinase and glucokinase is. The next enzyme that experiences a lot of allosteric regulation is phosphofructokinase 1, and in short, PFK1. This enzyme is negatively regulated by ATP and citrate. It makes sense why ATP is a negative feedback regulator because if there is a lot of ATP in the cell, then a lot of ATP will want to negatively feedback onto this enzyme and say, stop being very active because there's a lot of me. We don't need to do a lot of glycolysis. and that way it will reduce the activity of this enzyme. And citrate, we haven't gotten to it yet, but we will in this video. Citrate is a intermediate in the Krebs cycle, and because the Krebs cycle is more downward from glycolysis, it does negatively feedback onto phosphofructokinase, and if there's a lot of it, it says stop being very active because there is already a lot of me. Okay, there is also positive regulation going on through or by AMP and ADP, and this also makes sense in terms of if there's a lot of AMP and ADP, that means there is not a lot of ATP. So what AMP and ADP do, they feed forward, or sorry, not feed forward, they activate using allostery, they activate phosphofructokinase 1 and tell it to be more active to produce more ATP. The next the next molecule that does positive regulation on PFK1 is fructose 2,6-bisphosphate, and we will learn way more about this in the coming lectures, but to give you a little bit of foreshadowing, this molecule is regulated with glucagon, or regulated by glucagon and insulin, and it is active when those hormones are present, and consequently it activates PFK1. So more to come for this, but for now just know that it is positively regulating PFK1. And lastly, pyruvate kinase is the last enzyme in glycolysis. It produces pyruvate, and it is negatively regulated by ATP. Again, makes sense. If there's a lot of ATP, then we don't want this to work as hard. It is also negatively regulated by acetyl-CoA, and that also makes sense because a little later into this video, we will talk about how acetyl-CoA is formed right after glycolysis is over, and also fatty acids. You will learn this much later, I believe after the midterm, if I'm not mistaken, but fatty acids are long-chain carboxylic acids, and what they do is they produce acetyl groups in the forms of acetyl-CoA. So if we have a lot of fatty acids, then we will have a lot of acetyl-CoA, and that is how it will negatively feedback onto pyruvate kinase, telling it to stop its activity or at least reduce its activity so we don't make any more of these. And pyruvate kinase is also positively regulated by fructose 1,6-bisphosphate, and this is an example of feed-forward reaction, because if you remember your glycolysis, fructose 1,6-bisphosphate is in the beginning of the glycolysis pathway, whereas pyruvate kinase is the last enzyme. So if there is a lot of fructose 1,6-bisphosphate, then it will feed forward to pyruvate kinase and say, hello, come on, start working more, right? Like raise your activity because there's a lot of me and I need to get processed. So that is positive regulation. And I tried to give you a quick explanation on positive and negative regulation because it may be hard to understand this. I did try to look for a cartoon to explain it, but I couldn't, so hopefully my artistic skills help you understand whatever skills I have to help you understand positive and negative regulation. So if you think about having a conveyor belt, in all these three scenarios, we have a conveyor belt, and this person one is putting these gifts, these pink gifts on the conveyor belt, and the gifts are moving all the way to here to person two, and person two grabs them and puts them over here. Now this, we can think of this as the normal situation, right? The rate of this person putting the gift on here is the same rate as this person putting the gift over here. So nothing's backed up, nobody's sad, everybody's good. But what happens if, for some reason, person 2 is backed up with being able to process the gifts. Now this person is sad. And what happens is this person will negatively feedback onto person 1 and say, hey, stop doing this because I need time to take care of all of this. So the activity of person 1 will go down and give some time for person 2 to take care of all that it has before this activity can go up. So this is a case of negative feedback. What about feed-forward feedback? In this scenario where we have not enough at the end over here, this person is working too slow, and so this person isn't getting any gifts, and they're not being able to do their job, so they're sad again. And what happens is this person will regulate this person by saying, up your activity because this is too slow. Okay? And that's positive regulation. All right, to move on, the pentose phosphate pathway. There are two pathways in, it's short for PPP, pentose phosphate pathway, oxidative and non-oxidative. Let's begin with oxidative. Over here, I've, stitched up the pathway from the separate portions that Professor Hampton presented for your convenience. So in this pathway, we begin with glucose 6-phosphate, and the enzyme glucose 6-phosphate dehydrogenase oxidizes this molecule using NADP. And because NADP oxidizes the alcohol of glucose 6-phosphate into a carbonyl, NADP is being reduced into NADPH. This molecule is now called a lactone from organic chemistry. If you remember, a cyclic ester, which is what this is, right? We have our carbonyl, we have the oxygen. This is a terribly wrong depiction of an ester. It makes it seem like these are carbons, and they're not, right? This is just one bond to the oxygen. Oh, this is one bond to the oxygen. Then what happens is this lactone gets processed, if you will, by the enzyme lactamase, brings in a water, which cleaves this lactone bond. And now we are left with a carboxylate on one end, and this molecule is called 6-phosphogluconate. The next step that takes place is through the enzyme 6-phosphogluconate dehydrogenase. Again, we have the word dehydrogenase in the enzyme, meaning some kind of oxidation and reduction is taking place. And you would be right. NADP is oxidizing this molecule, and it is being reduced in the process. And also there is a decarboxylation. As you can see, this blue carboxyl is cleaved off in the form of CO2. Second or last step, we get ribulose 5-phosphate, and the last enzyme is a phosphopentose isomerase. And as you remember, isomerases just form the isomer of your initial molecule, so not too much changes, except we have a ketone over here, which turns into an aldehyde. And this is ribose 5-phosphate. And ribose 5-phosphate is later used to make many other molecules like nucleic acids, and yes, and this is an anabolic pathway. So we are building something instead of breaking it down. So that was the oxidative pathway. And now quickly let's talk about the non-oxidative pathway. And Professor Hampton didn't go too detailed about the non-oxidative pathway from lecture. What it seemed like all you should know is that there are this reaction happens to make a variety of different types of sugars that the cell can use at any point in time. There are these enzymes called transketolases and transaldolases, which play on moving around ketones or ketols and aldehydes to form these different types of sugars. And that concludes a recap of lecture 6. Now let's go into lecture 7. Well, I guess the big question in lecture 7 is what happens after glycolysis, right? So we've made pyruvate. Now what happens is, to be able to go into the Krebs cycle, pyruvate must turn into acetyl-CoA. And this reaction happens through pyruvate dehydrogenase complex. Now this is a complex and it has 3 catalytic subunits in it. And all of these 3 will in turn use an NAD, TPP, FAD, lipoate, and turn pyruvate into acetyl-CoA. So let's take a closer look into what happens exactly in this PDH complex. So what takes place is pyruvate is first decarboxylated by TPP. If you remember from earlier in this video, TPP attacks carbonyls and it either decarboxylates them or it can do other things also. It basically activates the carbonyl that it attacks and modifies the molecule in different ways. In this case, it modifies pyruvate by decarboxylating it. After this takes place, this acetyl group is still attached to TPP. Then lipoic acid, which is, or lipoate, which is this structure, or at least this is the business end of it. Right now, if you look at it, this is its oxidized form because we have the disulfide bond. But what happens is the oxidized disulfide bond of lipoate or lipoic acid becomes reduced, so it turns into 2 thiols, and one of the thiols grabs this acetyl moiety from TPP, and TPP goes back to being its initial self. Then what happens? A CoA comes in, and if you didn't get a chance to watch the lecture yet, please do, because Professor Hampton explains very clearly on this thiol. It's basically a part of a big molecule called coenzyme A, and at the very end of it, we have a thiol that is basically the functional part of this whole molecule. So what this thiol does is it steals this acetyl part from lipoate, and as it does that, CoA turns into acetyl-CoA, right, which is exactly what we wanted. And in the meantime, the reduced lipoic acid is still reduced, right? It's still in its thiol form, but FAD comes around, oxidizes the thiols into the disulfide bond, and FAD itself becomes reduced, but FAD doesn't stay reduced, right? It doesn't stay as FADH2 because NAD comes around, oxidizes FADH2, turns into NADH. So at the end of this whole catalytic process, make sure to be able to walk through all of these steps, right? From pyruvate to PPP to lipoate to CoA to the oxidation of lipoate and the fact that the oxidation happens because of FAD, and then NAD returns FADH2 into FAD. At the end, we're left with a carbon dioxide, an acetyl-CoA molecule, and NADH plus H+. Also remember that the three catalytic subunits we talked about, E1, E2, E3, it would be useful to remember their names, and I believe they are color-coded over here. So this is 1, this is 2, and this is 3. Okay. So after this reaction has taken place, we have now accumulated the acetyl-CoA needed to shunt to the Krebs cycle. So let's talk about the Krebs cycle. Acetyl-CoA enters the cycle, and the first step is catalyzed by citrate synthase. And what citrate synthase does is it takes the molecule of oxaloacetate and it combines it with acetyl-CoA to form the molecule of citrate. In the meantime, a water molecule is added and the coenzyme A is kicked off. The next step that happens is citrate becomes isocitrate, but we need to talk about the isomerization that takes place in between that reaction. And this reaction is catalyzed by aconitase. And what happens is this OH over here is simply moved to, it was on this carbon and the middle carbon, and it is moved to the bottom carbon. And what you will always notice is that in the first round of Krebs, the OH is always moved on the carbon that does not belong to the newly entered acetyl-CoA. If that doesn't make sense, I'll say it again. Acetyl-CoA comes in. This is the acetyl portion of acetyl-CoA, and it is color-coded for you. This is that portion, right? It continues to be in these molecules over here. So this OH of citrate that resides on the middle carbon is always moved to this bottom carbon and never this carbon. And that just happens because the enzyme aconitase has that specific structure where it only catches citrate in a specific way where it can only catalyze the movement of OH onto this carbon and no other carbon. Okay, now we have isocitrate. Isocitrate goes through a process called oxidative decarboxylation through the enzyme isocitrate dehydrogenase. And again, if there's a dehydrogenase in the enzyme name, that means that something is being oxidized and reduced. And in this case, you would be correct if you thought that, because this reaction produces an NADH because it initially used an NAD to oxidize isocitrate. And as this reaction's name is oxidative decarboxylation, we have also got rid of one carbon in the form of carbon dioxide. So now we are left with the molecule alpha-ketoglutarate. Alpha-ketoglutarate goes through the reaction that is catalyzed by alpha-ketoglutarate dehydrogenase complex. And this complex is, it works exactly the same way as pyruvate dehydrogenase. It uses a coenzyme A, TPP, FAD, lipoate. So this mechanism is the same as what we talked about earlier in this video. Another carbon dioxide is liberated from alpha-ketoglutarate. So now we are left with succinyl-CoA. Succinyl-CoA is catalyzed. This reaction is catalyzed by succinyl-CoA synthetase. And another tip for you, if you have the word synthetase with a T in it, always know that most of the time you will be producing or using an ATP of some sort, right? So you can either have a synthetase or a synthase if you look over here up at the top. See, synthase doesn't have an ATP involved. However, synthetase, because it has the T, it's a good way to remember that an ATP will be involved. In this reaction, GDP is turned into GTP. However, it doesn't stay GTP. This model doesn't show it, but what happens is GTP, ADP steals a phosphate from GTP, which goes back to being GDP, and the ADP that came in over here turns into ATP, and that's what the cell keeps. The next reaction uses succinate and turns it into fumarate, and this is basically another dehydrogenation reaction. Again, the enzyme is succinate dehydrogenase. It has that dehydrogenase name in it, meaning something is being oxidized or reduced. And in this case, FAD is being reduced into FADH2 as it oxidizes succinate into fumarate. The next reaction is catalyzed by fumarase, and what fumarase does is it brings in a water molecule and it hydrates fumarate into malate. And the last reaction in the citric acid cycle, or the Krebs cycle, is catalyzed by malate dehydrogenase. Again, an NAD is used to oxidize malate. In this case, it oxidizes this alcohol and forms this carbonyl, and an NADH is produced. And there we go. At the end, we produce an oxaloacetate, which goes back in to the beginning, air quotes, beginning of the citrate of the Krebs cycle, and again gets together with an acetyl-CoA, and the cycle goes on and on. An important thing to mention is, per glycolysis pathway, we produce 2 molecules of pyruvate, right? Meaning for every molecule of glucose, we produce 2 molecules of pyruvate, and each of those molecules will go through the Krebs cycle. And that means that per molecule of glucose, we will have 2 of these Krebs cycles. That's why earlier I said during the first round of the cycle, the acetyl-CoA, the acetyl portion of acetyl-CoA kind of stays intact, and then and this hydrogen isn't added to this carbon, right? So that's the Krebs cycle, and I've also found this more simple version of the Krebs cycle if you are a person who doesn't prefer the carbons and hydrogens showing. You can take a look at this diagram or this pathway, and it also has the enzyme names and everything else blank, so maybe you can use this as a blank sheet for you to fill out and practice. But basically, these molecules are portrayed in a more organic chemistry style, where the angles are carbons and the lines are bonds. And with that, this is the end of this video. To remind you, please, if you have not read the writings that Professor Hampton has posted, you should, because they will give you a good foundation of what we're talking about in lecture and in discussion. So If you haven't read those, stop this video, go read them, dissect them. They're easy to read, but they have a lot of useful information. And of course, if you have any questions, my name is Lilith. Please don't hesitate to email me or visit my office hours on Mondays at 7 p.m. I will post this video on my pages on Canvas. If you scroll all the way down below, on the Canvas homepage, every IATA has their own page, and I'll post these and my notes on there. Good luck, keep studying, and you got this. Bye.